Lalani, and I'm a member of the Aga Khan Health Board, and I'm on the non-communicable disease portfolio. It's my pleasure and privilege to introduce our breakout session and our breakout speaker today. It's a topic that is very interesting. It's been very topical for a while now, but something I think is still quite poorly understood by even persons within the medical profession. I often get questions from friends, from members of the Jamaat, and unfortunately I'm not really able to answer them very well. So I'm really looking forward to today's session in the hope that I may come away with some greater understanding of stem cell research. So let me introduce now Professor Dr. El-Nasir Lalani. He's a professor of stem cell biology and translation medicine and a professor of molecular and cellular pathology at the Aga Khan University Center for Regenerative Medicine and Stem Cell Research in Karachi, Pakistan. He was appointed as founding director of the Stem Cell Research Unit in August 2016. And prior to that, he was the founding dean of research and graduate studies at AKU for 8 years. Prior to moving there, he held professorial positions at the University of Birmingham and Imperial College. And he has published over 200 papers. So without any further delay, can I hand over to you, Professor Lalani, to take us away, and you can share your screen, please.
Bismillahirrahmanirrahim, and Yali Madad, everybody. First of all, before I start, I would like to thank Badala, the chair of the Aga Khan Health Board, and all members of the Health Board and the organizing committee for giving me this opportunity to speak on a topic which I think is current and will remain current for many decades ahead. The field of stem cell biology and regenerative medicine, I think will have a paradigm shift, including the application of artificial intelligence in this area, which will probably lend itself from treatment to cures. So what you just heard, an excellent presentation by Professor Sir Munir Muhammad. I think if you take that in terms of us using therapies to treat cells, this is about using your own body cells to treat yourself. Today we are in the midst of a pandemic, and we know about it because of the lockdown that we won't experience globally and continue to experience in some parts of the world. The fact that every day there's not an hour that goes by that you don't have it on your news channel or on your mobile phone. And so people are aware of what SARS-CoV-2 and COVID-19 is because of the information that's out there. And so there's a pandemic going on, but there has been an invisible pandemic going on for many years. And then this relates to non-communicable diseases, which Chairman Akbadala spoke about. Now, you may know this, that SARS-CoV-2 or COVID-19, every 30 seconds a patient dies from COVID-19. Some of these deaths could be preventable by masking, physical distancing, and hand washing. And I would urge everybody that's listening in, please mask, keep your physical distance, and hand wash. We've had over 1 million deaths, 35 million people have been confirmed with the cases, and only 650 million people have been tested out of 7 billion. Now these figures are startling to you, but every day, or every 30 seconds a patient dies, or an individual dies from diseases that could be treated with organ replacement. So by the time we finish the seminars today, over 2 hours, 240 people will have died because they couldn't get an organ replacement. 240 will have died because of COVID-19. But inshallah for COVID, within a year or two, a bit longer, there may be a vaccine or even therapies that will be able to treat this particular virus. So organ transplantation currently only covers 10% of the global need. And most of the organs that we are transplanting are kidney, liver, heart, lung, heart valves, and the pancreas in that particular order. And as Chairman Allah said, every minute, 80 patients or individuals die of a non-communicable disease. In the 2-hour presentations today, 10,000 people globally will have died from a non-communicable disease. And these diseases are predominantly heart disease, cancer, diabetes, lung disease, etc. And these figures were mentioned earlier. 60 million people die globally every year. Of these, 40 million die from non-communicable diseases, 70% all deaths. Each year, 15 million of these individuals die between the age of 30 and 69. And 80% of these premature deaths occur in low middle income countries where the Aga Khan Development Network works. And as I've already mentioned, They die of what diseases? As you can see on the slide: cardiovascular disease, diabetes, chronic inflammatory diseases, and cancer, which make up nearly 84% of all diseases. You heard in Professor Sarmuddin Muhammad's presentation that a lot of these diseases occur with age. These are age-related diseases. So aging is a stem cell disease. And we accept aging graciously, and we say, well, we age and we accept it. But this question of why we age, and there's some biological reasons for this, but why do we age? And one of the reasons, one of the major contributors to aging is the number of stem cells. So when we are born, let's assume we had 100% stem cells, it decreases with age. Both the numbers of stem cells decrease, they may die, or they may even go into hibernation. So every time you put a candle next time on a cake, just remember that the number of stem cells that you have in your body has decreased since the year previously. Whilst you're celebrating your birthday, your stem cells have decreased significantly. So this whole area of non-communicable diseases, a lot of this has to do with the stem cells in your body, their ability to regenerate parts that are damaged. So every time you have an injury, your stem cells come into effect. So stem cells, aging are related. Aging is a stem cell disease, but there are many other factors that also contribute to aging. So what are these stem cells? Now, some of you will know a lot about this area, so please excuse me, but this is an important area just to bring everybody online. So the first thing is, when the sperm fertilizes the egg, you have the formation of the embryo. You start with a two-cell stage, goes to 4, 8, 16, 32. Eventually you form what's known as a blastocyst. And in the blastocyst, you have an outer membrane, or inner cell mass, and an outer cell mass. These cells together go on to form us. They form us in the question that the outer cell mass forms the placenta. And these cells inside, these are the embryonic stem cells, same as these ones here. These form the rest of us. Within 12 weeks, these cells go on to form the eyes, the ears, the brain, every single organ in the body, the legs, the skin, all the blood vessels, the nervous system, the immune system. But this is an area, those of you who are interested, we understand poorly, or not at all. We don't know which cells are going to come in towards what lineage, which cells are going to make the heart or the lung or the liver. How do they orientate? How do you make the right side of your body, the left side of your body, the top and the bottom, the orientation? None of this is as yet known. But all this fascinating feat occurs within 12 weeks in the first trimester. And you have these cells that I mentioned to you about, the pluripotent stem cells. These cells that are inside here, these are the embryonic stem cells. They have the ability to make every organ in the body. What they don't make is the placenta. However, there is recent evidence in the last 3 to 4 months which shows that it's possible in the lab to even take these cells and create at least one part or some of the cells in the placenta. So these embryonic stem cells, or pluripotent stem cells, have the ability to make all cell types in the body. We then go on to the next layer of complexity. So there's a hierarchy from totipotent stem cells, which can make all the cell types in the body and make you and me, including the placenta, pluripotent stem cells that can make all the organs, but would not be able to make us, and then multipotent stem cells that are what's known as lineage committed, the mesoderm, the endoderm, the ectoderm. And then each of the organs that we have a stem cells in those compartments, and they can only make those organs. So scientists going back around the time when Hazim Imam mentioned this, were thinking, is it possible, something you would have thought was a crazy idea, is it possible to take any one of these cells, including a skin cell, and take it back into time to make it into an embryonic stem cell type? So the question they were asking was, is biological time travel possible? And the answer is absolutely. This has been demonstrated. So in 2006, Shinya Yamanaka, Professor Shinya Yamanaka, working at the University of California in San Francisco at the Gladstone Institute, and we collaborate and we have a partnership with UCSF on our stem cell program, did exactly that. He managed to take skin cells into the lab and added 4 factors. Nowadays, you can do it in many other ways, added 4 factors, and uses a virus, and we use viruses as well, but you can use other agents to get them into the skin cell. And then, I'm just going to use, it's not a scientific term, but magically, things happen, and these cells are converted back into time. So they've time traveled back to becoming similar to those embryonic stem cells. Because they are made in the lab and we have reprogrammed them, they are known as induced pluripotent stem cells. So today, in our lab here at AKU and in many labs in the world, we can take a skin biopsy from any one of you or a blood cell, take it into our labs, add some factors to it, and convert it into an iPSC cell. This cell will have a memory, by and large, to the cell that was in when you were in your mother's womb. We can then add other factors to it and convert it to any cell type in your body, a heart cell, a liver cell, a kidney cell, including now a sperm and an egg. Now, we can then transfer these cells back to your body where it is required, whether it be for repair processes or for other purposes. The other thing we can now do is genetically manipulate that by a technique known as CRISPR-Cas9. There are other techniques that are coming forward. Remove the defective gene, insert it in one of these cells, and put it back into your body. We can study diseases. You heard Professor Primama talk about precision medicine. But what we can do here is somebody has an arrhythmia, you saw on that ECG being normal, but assuming there was an abnormal ECG and it was an inherited deficiency, we could grow the cell in the lab, convert it into a cardiac cell, and then test the drug that would change that into normal rhythm and reimplant it back into the patient. A note of caution, these cells also have the ability to form a tumor. And I can talk about that when we have question and answers as to what's the implication of that. So what's regenerative medicine really? Regenerative medicine is about regenerating tissues, replacing tissues, or repairing tissues. Now, we regenerate on a daily basis. Our skin regenerates, our gastrointestinal tract regenerates, our hair, those of us who have hair follicles continue to produce hairs, the bone marrow regenerates. So regeneration is part of a normal cycle that occurs in us. It occurs in most tissues. In a case where if a liver is removed, 2/3 of my liver were to be removed, within 4 to 6 weeks, it could be fully replaced. So every organ in the body has that capacity, but that capacity is dependent on residual stem cells within that organ. I'm going to illustrate an example of what happens. This is an ability we still have, but not to the extent that you might see this in amphibians or fish. So I'm just going to put this video on. And what we have done here is, this is a newt where a limb has been sectioned and time-lapse photography is now taken. And you can see the limb regenerate. It regenerates to the right size, has all the same features as it would have otherwise. Now this is a cartoon, and in the cartoon, it's wishing us, Ya Ali Mother, the greeting us to start of this evening. And it's going to go through a section, and it'll just show you what happens. Now, this is a very complex process that occurs over several weeks. So once the limb has been severed, you have to have stem cells that come together. Stem cells that are going to make bone, cartilage, muscle, skin, nerves, blood vessels, all have to come together in a coordinated manner to form this limb. The limb has to be of the right size. It has to be angular. It has to have sensation. And so you can see the different colored stem cells that go on to form this. Do we have the ability of this? Of course, we do have the ability, but our ability decreases as we age. And the idea now is, can we, using stem cell, understanding the biology of stem cells, do the same with our organs? Now, this is an example of regeneration. This is a 66-year-old lady at another institution. She has got a diabetic ulcer. She had been treated for many, many years, but now the only opportunity that was left was that she was going to get a below knee amputation. What colleagues did here was not use stem cells, but use what's known as extracellular matrix. These are a mesh that interacts with your cells. And the question here was, can we stimulate the stem cells that are surrounding this particular wound at the periphery to stimulate it for the skin to grow over this? And this is what happened after 11 weeks. There were no stem cells used here. These are giving stimuli using extracellular matrix. We put on this on the surface, and here you can see that the wound has been healed. Now, the implications of these are far-reaching, as you can imagine, not only for diabetic ulcers, but ulcers that occur for many other reasons, and also wound healing. What about making organs? So this is from colleagues at Wake Forest, and I'm just going to play this video.
Grow your own organs in the lab means no rejection problems and no waiting around for organ donors. It's called regenerative medicine, and it's an exciting future that awaits us all.
I see us getting to a point of having options available that actually stop disease processes, reverse disease processes, or offer people cures.
Simple organs, like bladders, are the easiest to grow.
And we then create a three-dimensional mold. And we place the cells on top of the mold.
The mold is in the shape of a bladder and is made from material that breaks down in the body.
We place the mold with the cells in an oven-like device. We cooked it, if you will, very much like baking a layer cake. And we then were able to take that organ out and we were able to place it into patients. Another of the organs that we have targeted is the urethra, which is the channel that connects the bladder to the outside of the body. It is a very important organ, as you can imagine.
Some of Anthony's patients had had car accidents, damaging their urethras. So he decided to grow them new ones. They were like bladders, really, with a different geometry. Scaffolding was seeded with the patient's cells and nutrients, and then sewn into the shape of a urethra.
And we then were able to place those engineered urethras back into those patients.
To automate organ making, the researchers came up with an incredible method. Print them. They even started out with modified computer printers.
But instead of using ink, we use cells, and we print the cells with a gel-like material, one layer at a time, and we then allow the gel to get harder over time.
They're developing another application for bioprinting, for wounds.
One of the strategies is to have a printing machine that not only prints, but also scans. So basically, the patient is first scanned, so the wound area gets a scan of the wound, and then we're able to go back with the printer and print the right layers of tissues right where they belong.
Now, for most organs, there's still a long way to go before they'll be ready for patients. But research is progressing on artificial kidneys, heart valves, large blood vessels, and skin. Here it's being slowly stretched out. They're even working on artificial ears and fingers. Predicting the timing for any research is always difficult, but it's clear some pretty exciting developments are on their way.
The other thing we can do with iPSC cells is we can make mini organs. Now the term organ, I need to be careful here. People talk about mini brains, mini kidneys, mini lungs. These are not the complete organ, but there are parts of it. So for example, at Aga Khan University here in our lab, we have grown brain organoids, but these are cortical organoids. Just the area that you can see with my cursor, with the ventricular zone here, which has got the stem cells. We've been growing these, and we can grow these for 40 to 50 weeks to study brain development and also study neurodevelopment in the brain. We are working on autism spectrum disorders and on particularly one degenerative disease by manipulating genes, knocking particular genes out to see what happens here and studying that. For SARS-CoV-2, a lot of interest in the stem cell field is the one that's really leading to understand how the virus enters particular cell types in the lungs. So this is called the many lung, but it's not just particular epithelium in the lung. And there are type 2 pneumocytes, the special cells in the lungs that the SARS-CoV-2 virus has a predilection for, as it goes to S2 receptors. So the virus not only affects the lung, it affects the heart, it affects the kidney, it affects the pancreas. And we can develop in our labs, and these have been developed through stem technology, these so-called mini organoids. Any particular organ can be developed, and then we see how the virus gets inside the cell, and how can we, using the methodologies that Professor Pirmama talked about, how can we do that to combat the virus? The other thing we can do in the lab, the other question that comes up is, where are we going to get these organs for transplantation? Ideally, we would like these organs to come from ourselves, for us to be able to donate these, or from cadaveric transplantation. But as you saw from the earlier slide, supply and demand is a challenge, and only currently 150,000 transplants are done worldwide, and we need at least 1 million transplants a year. So one area is, can we do this in animals? And we need to have the organs growing of the right size to be able to transplant into humans. And there are two animals that this can be done in macaques and in pigs. Okay, and here is an example. So here's a rat with the skin that has come from a mouse, and the mouse has got rat skin. And this is a mouse in which there is a degree of human cortical tissue. I wonder what this mouse was thinking about. But this raises many ethical questions. When you humanize an animal, does that animal have human rights? A question that I'm sure we will continue to discuss, and ethicists will continue to discuss to answer the morality of doing this. And lastly, very much more recently, over the last 3 years, we've been able to do something which has got implications for man's evolution. That is to be able to take skin cells and convert a skin cell from any individual into an egg and a sperm. This is called gametogenesis. And the potential for this is that it could replace in vitro fertilization. We could have thousands of eggs made and thousands of embryos. We could screen for the best embryo, and using the technique, as I mentioned, of CRISPR-Cas, we could enhance height or memory or intelligence or eye color. That takes us into the area of eugenics, and it'll be interesting to hear your views when we have the question and answer. But importantly, this could influence our evolution and future generations. So as I mentioned, 15 years ago when Hazrat Imam talked about this particular area, I don't think anybody could have guessed that this is where we would have been 15 years hence. 15 years on from now, well, your guess is as good as mine, but I think we will have number of diseases that will be treated through stem cell therapies. Organs may take a little longer. Now, this particular slide shows the kind of trials that are ongoing. The green ones are ones that are in phase 3. The yellow ones are in phase 2, phase 1 and phase 2, and the organ ones are somewhere down the line. There are over 1,000 companies working on this. $8 billion have been invested in this particular area. And there are currently 1,500 trials ongoing just on stem cell, including 35 to do with COVID-19. I'm going to leave you with this last presentation. This is from from Stanford University. This is to demonstrate, we don't understand the science of this, but the potential of stem cell therapy.
For the longest time, it was thought that brain damage, such as that caused by a stroke, is permanent and irreversible. But a recent medical breakthrough is bringing this notion into question. Researchers at Stanford University have been blown away by the results of a clinical trial they conducted.
5 years ago, Sonia Kuntz suffered a stroke that severely damaged her brain.
This is Sonia's stroke.
It partially paralyzed Sonia on her right side, and she could barely speak.
Her speech was not very understandable. She couldn't order food or communicate well.
2 years later, Sonia could still hardly lift her arm. But just one day after an experimental treatment, oh my gosh, Sonia could lift her arm over her head. Can you move it to the side? And move it to the side. And also to the front. And her words began to flow.
I woke up and immediately I could speak better.
Dr. Gary Steinberg, chair of neurosurgery at Stanford, led the small clinical trial. 18 chronic stroke patients were involved. 12 came to Stanford, including Sonia.
I was very excited. I think I started to cry.
In the trial, Steinberg drilled a tiny hole into the patient's skull and using a very fine needle, injected modified human adult stem cells around the stroke.
We put them around the stroke, and that's where they do their thing to recover the function.
These cells don't survive for long after the surgery, but they appear to trigger a patient's damaged brain to begin to heal itself.
The cells somehow jumpstart the system again.
Here's another stroke patient just before surgery. And here she is just days after receiving the stem cell treatment. As for Sonia, her life is back on track. She's now married and pregnant with her first child.
And it's a boy. Yeah.
This could revolutionize our concept of what happens, not only in stroke, but traumatic brain injury and every neurodegenerative disorder. We thought the brain circuits were dead, and we've learned that they're not. End quote.
So finally, we're entering a brave new world where through stem cell technology, we can make synthetic gametes, synthetic embryos, we can underdo gene editing, influence human development, and if you add to that artificial intelligence, we are now in the control of evolution. This raises many ethical questions, but this is where the area is going to be going over the next 50 years. But it raises many questions that society needs to answer, allowing for the scientists to be able to take this forward. Thank you for your attention.
Thank you very much, Professor Lalani. I have some specific questions from members of the Jamaat with regards to stem cell therapy for specific disease conditions. So the ones that seem to be the most popular, so ALS, amyotrophic lateral sclerosis or motor neuron disease, and also osteoarthritis of the knee. Is there any way we can reproduce cartilage to treat this?
Okay, so I think those are questions that come to me probably on a regular basis. Regarding osteoarthritis, I think you will hear many people who have gone to clinics where there is no data to support what these clinics do. Remember, once, unlike what Professor Pir Mahmood mentioned here, unlike taking a drug, you can stop a drug. When you have cells that are injected into you, you can't remove them once they're in there. So you will hear people say for osteoarthritis and other arthritic diseases, we will inject cells into your knee and you will feel better. Now these cells are taken from where? They're either taken from fat or bone marrow derived mesenchymal stem cells. Then usually these cells go on to form cartilage, bone or fat. They can also form tumor. We do not know at the present time how many cells are required, whether they will go and heal the cartilage, what will happen over a period of time. There are trials ongoing, so we await to see that. But please be aware of the thousands of clinics that are offering bogus treatment based on evidence that does not exist. Big challenge that we face globally. About ALS, the same thing. There are trials ongoing. Watch this space, but it's still early days. They're more like phase 1 trials in some areas that are being undertaken. The danger here is the clinics that are offering, both in the United States, in Europe, in China, in India, treatments that are unproven. And this is one of the biggest challenges the stem cell field has encountered. If you are interested in going into trials, please do write to me. I will guide you where you need to go and check and see if you can enroll in trials at A1 centers, which is also backed up by science.
Thank you, Professor Lalani. We don't have much time, but there is a question, and I'm not sure you'll be able to answer it in detail so quickly, but for gametogenesis, what ethical considerations are taken into account? And that's from Farnaz Jamal. Okay,
so first of all, for gametogenesis, first of all, the fact that through this particular science we can do it. The question that arises is the following. It happened when we started in vitro fertilization. And this is now over 40 years old. We have now got 6 million test tube babies. It's an experiment that's ongoing. So the question here is, if we can do this, and then do we need to do in vitro fertilization? For parents or individuals that are going through this, they don't have any children, here's some hope it offers, that through this particular technology, if you're unable through IVF, this is a technology that could offer you the opportunity to have children. However, the challenge here is that now we are getting to a place whereby we can influence the future generation. This is about eugenics, where you can take away genes. So for example, if somebody has an inherited disease, I think there'll be no argument amongst most of us to say, if you can replace that particular gene in the gametes, then your offspring will not have that disease. However, when it comes to enhancement of any kind, raises a big question. And if you raise this, people say, no, we wouldn't do it. But if you had the chance and say, listen, you can go away to a particular part and there is a professor sitting there willing to do this, I wonder how many would not take that chance if they could afford it. But it is unethical to do so at the present time. And I'm not personally in favor of any of these eugenics ideas, apart from the fact that if you can replace genes that are faulty and that are inherited, that's an area that I think much work will be done in the future.
Many thank you to Professor Lalani and to everyone also for joining today.